That one. Okay. So, good afternoon, everybody, and welcome to a very, very special Grand Rounds. This is a combined Grand Rounds with the Global Health Grand Rounds and Internal Medicine Grand Rounds. Uh, Global Health Grand Rounds are sponsored by University Hospitals Royal Green Center for Travel Medicine and Global Health, and Global Health Grand Rounds occur virtually the second Tuesday of every month. And as kicking things off, we're combining this with our usual every Tuesday at noon Department of Medicine Grand Rounds. And there could be no more appropriate speaker than Dr. Ofotokun, who, of course, in Department of Medicine, known as our chair, who's been here full time since October 1st. It seems like you've been here a while and already having a huge impact. Dr. Ofotokun, before coming to UH, was the Grady Distinguished Professor of Medicine at Emory for many, many years, Associate Dean for Research at Emory School of Medicine, co-director of the Emory Center for AIDS Research Center, has had a major impact on the global stage for many, many years. Activities in Nigeria, Ethiopia, Georgia, Vietnam, incredible academic career, 250 peer-reviewed publications, has received two big awards from the Infectious Disease Society of America, the John Injers Award, the Maxwell Finwell Award, is an elected member of the National Academy of Medicine, and recipient American Academy of Microbiology Honorary Diversity Lecture Award. So we're thrilled to have you here at Case Western, UH, the VA, Global Health Grand Rounds. Thank you, Igho, very much for kicking us off.
Thank you. Thank you so much. Is it possible to take this?
I just need to share.
Yes.
No, I got it.
All right. Awesome. Thank you so much, Dr. Armitage. It's really a pleasure to be here. And I'm going to take the prerogative of the chair to make a few announcements. One, Grand Rounds are very important to the Department of Medicine. It's where we come together on a weekly basis. You know, it's like in our academic rendezvous. And I want to encourage all of you, every member of the Department of Medicine, regardless of division, faculty, fellows, students, residents, I really want to encourage you that you show up for this Grand Round on a weekly basis. It's an opportunity for our residents and fellows to mingle with our faculty, have presentation and have opportunity to ask questions. And so I really, really want to put a big plug for attendance of Grand Round. Dr. Armitage and his team do a lot of job. It's a lot of work to put this together. and it will be great to see everybody. And we will use it as an opportunity to occasionally update all of us about what is happening in the department. So Dr. Armitage, in his role as the executive vice chair for the department, will use it also as an opportunity just to give important information that might have an impact on all of us. So I really, really want to encourage participation in Grand Round going forward. Really privileged to be giving this lecture. I have titled it From Domestic Health to Global Impact, Advancing Innovation in Biomedical Research. First, we can talk about global health here at UH, Case Western, without acknowledging the contribution of Bob Salata and Ro Green, two incredible leaders of our global health effort. For many, many years, Bob Salata championed, the effort to really bring our global health program initiative to the forefront, and supported by his friend, Ro Green, who not only support global health with her resources, but with her passion. And one of the things that I really love about what we see here in Cleveland, it's the community endorsing our program with their philanthropic contribution. So I see philanthropy, the dollar is important, But the significance of philanthropy is that we are aligned with the value of our community. So Roe Green, the Tibbs family, the Salata family, all contributing towards our global health effort. And I also want to thank the Health Services Research Center team, Sam Bora and his team in the Health Services Research Center for this idea of having a Roe Green travel, medicine, and global health um, grand round. I think it's a commendable effort. And we hope that we can continue to use this to coalesce our global health efforts together and to highlight the contribution of our community to our global health effort. So two things I want to do today, just to kind of, the first two parts of this lecture, I want to just talk about how we build our research capacity here in the United States, our domestic research work. And then the second half, which is the more important part, is how we leverage our domestic resources to support global health, to support global health impact. So let's begin with the first half. So I started my career when the AIDS epidemic was breaking. So this was back in Africa. And I think almost everywhere in the world, people at the beginning of the epidemic, Usha, who is looking at me, or the ID physician, Dr. Armitage, we knew that almost everybody died. AIDS was a death sentence. And so this is what is shown here in this. This is data from the Danish group, where you look at before the introduction of treatment, everybody died. Mortality rate was just near close to 80, 90%. People got AIDS, they died. And then around towards the early 1990s, 94, 95, 96, the treatment came. And what we saw was saw a bent in the epidemic. Survival rate increased significantly. Initially, it was there was a big bump, and then it got better. Now, if we were to do the same study, you find out that people living with HIV now have a survival rate that is almost close to the general population. And so this is really reflected in Africa, South Africa. This is data from KwaZulu-Natal, a small community in South Africa where the epidemic was just massive. During the peak of the epidemic, life expectancy dropped significantly below 50 years as a result of the epidemic. Then around 2004, treatment came to Africa, and you can see a significant impact of treatment on the epidemic. And so this, on this left side of the slide, you can see here just the progress that we made. No treatment, toxic treatment, much better treatment. By the time I started as a faculty in 2003 at Emory, we've gotten the treatment of HIV down to 1 pill once a day. You know, as a resident, I spent a lot of my time not only managing the disease, but managing the toxicity of the disease. And then what happens? People were living longer. And then we started thinking of, oh, there might be much more to this epidemic. Not only did we save life, we were not able to completely eradicate the virus from the body. The virus stays, people take medication every day. So the question is, what is the impact of living with a virus and taking therapy for the rest of your life? So if you've heard me give a talk, I believe in the doctor-patient interaction as the driver of innovation in biomedical research. And almost every manuscript that I have written, almost every grant that I have written, can be named to an encounter with a patient. You know, an encounter in the clinic, an encounter in the hospital. And so this was a generic picture, but really to remind you of this encounter that I had around 2005. I was relatively new at Emory, and I inherited a panel of HIV patients. And so this woman who came to my clinic had been living with HIV for at least 20-something years. And she also has a daughter, a young adult, who was my patient. So her daughter was perinatally infected from the mother. So the mother had gone through series of treatment. There was no treatment, then there was treatment, toxic treatment, developed a lot of drug resistance. So I was struggling to get her suppressed, changing regimen to regimen until eventually we were able to get her viral load suppressed. Then one Wednesday, she came to clinic and said, Dr. O, on Sunday, I bumped my foot against the doorpost. That was 2 Sundays before. And this has been hurting, it's been paining. And I looked at the foot, it was not red, it didn't look like there was infection. If I want to see it, maybe it was slightly swollen. And since she said it was paining, it was hurting, I said, okay, go get an x-ray. And so she went down, got an x-ray. And then she came back and she has this hairline fracture in one of her metatarsal bone. I was like, no big deal. Go to the orthopedic. They gave her the usual boot and she wore the boot for 6 weeks. Swelling went down, pain went away, bone healed. And I thought, well, that is the end of the story. About 6 months later, she came back again, another fracture. And 6 months after then, another fracture. Within a period of 2 years, she's had at least 3 to 4 very minimally provoked fracture. So that got me thinking. So this is what we do in academic medicine. We not only see the patient in front of us, we want the patient in front of us to inform us how to take care of the next patient better. Curiosity. Why would a young Black woman in her mid-30s have fracture consistently, 3 fractures in a period of less than 2 years? I just kept thinking about it. Just kept recurring and recurring. This doesn't make sense. In the first place, Blacks generally don't fracture. We have thick, very dense bone, especially Black men. Fracture rate is very uncommon. And for a pre-menopausal Black woman to have 3 fractures. And by then, nobody really knew the connection between HIV and end organ damage. And so that got me thinking, could this have anything to do with her drugs? Could this have anything to do with her HIV infection? And so I went into the literature. And the only 2 papers that I could find about HIV and bone disease were this paper, at that time from WashU. Bill Padley, who is a friend of mine, was the chief of ID at WashU for many, many years, and Pablo Teblas, who was then a trainee, a protege of Bill Padley, was at WashU. Now he's at UPenn. So had these 2 papers. They said, well, I told them, why did you publish this paper? They said, well, we were actually looking at fat accumulation, lipodystrophy. When you give people some of the HIV medication, the protease inhibitor, you have abnormal accumulation of fats. So they were doing DEXA scan to just study lipodystrophy. They had series of DEXA scan they have done. And then one of the nurses just drew their attention to the data on the DEXA scan and said, look, we're looking at lipodystrophy, but this patient have osteopenia and osteoporosis. And that was how they published these 2 papers. And it turned out that the nurse was actually a very observant nurse. Later, it turned out that osteoporosis, osteopenia was very common in people living with HIV, that 2 out of every 3, at least 15% are frankly osteoporotic, and another 50%. So is our osteopenia. That is 2 out of every 3 individuals living with HIV have some form of abnormalities in their bone. Not only that, it's good. So you have osteopenia, you have osteoporosis. Later, Virginia Tran at Mass General looked at the rate of fracture. So this weakness, this thin bone, was leading to increased rate of fracture in this population. You can see here, this is data from Massachusetts. Female, you see the rate of fracture significantly higher in women living with HIV. Men in particular, the protection that men have as a result of their dense bone was completely wiped away. You see, men usually don't fracture until they get to around 65, 70. You can see as early as 30 years, third decade of life, you had high body incidence on fracture. And this is data from Europe, where you see hip fracture was about 9-fold higher in people living with HIV compared to the general population. And so this really got us thinking, why would people living with HIV have osteoporotic bone? So this is what it looks like. This is healthy bone here on the left, dense, well mineralized, and you see what HIV does to the bone. Chews up the bone. You see porous, loss of mineralization. And it turns out that this is not a phenomenon that is unique just to the bone. We see that cardiovascular disease, MI, was more common in people living with HIV. And this is the data from Lorraine Collins, who was my fellow for a long time now. She's was my colleague, now an associate professor, highly well-funded, and we were looking at the prevalence of end organ damage, 10 different comorbidity, aging-related comorbidity. We found that regardless of every organ that you look at, you see that HIV really predisposes people to end organ damage. And so this really led to this series of work that we did, where we started to look at the impact of HIV on aging-related comorbidities or end organ. So we focus on the bone, the heart, and the brain. And I am going to just share briefly with you our work on the bone, just to kind of highlight how we build our capacity domestically and how that capacity then help us to do some of the global work that we are doing. So when I started out asking questions about the bone, my knowledge I wasn't a bone biologist. I had a colleague who is a collaborator, who is a bone biologist. All I remembered about bone was what I was taught in medical school, histology class. My histology teacher, was Dr. Rao from India. I still remember his voice talking about osteoclast and osteoblast. So it turns out that when we're born, we have a rapid growth in our bone until around the age of 25. And then the bone mass peaks. And then we maintain what we call the peak bone mass between the age of 25 and age of 55. And so during this period, we go into what we call remodeling. So what that means is that when the bone is old, it's broken down, and then new ones are formed to replace them. And all of us, during this period of 25 to about 60, we replace our bone, our entire bone, every 10 years. So you actually have new sets of skeleton every 10 years. It turns out that the two cells that do this are the osteoclast, which is responsible for breaking down old, worn-out bone, and then osteoblast, which forms new bone to replace the ones that are broken down. So the question, therefore, was, what does HIV have to do with osteoclast and osteoblast? So it turns out that osteoclasts, the cells that are responsible for breaking down of bone resorption, are cells of the immune system. They are cells of monocytic lineage. So you recognize them because they have this receptor, RANK, receptor activator of NF-kappa B, on their cell surface. So the RANK has a very potent ligand, RANK ligand, receptor activator of NF-kappa B ligand. So what happens is that this ligand binds to the receptor on the surface of osteoclast precursor, induces these cells to proliferate into what is known as pre-osteoclast. The pre-osteoclast then fuse together to form these more giant multinucleated cells that are responsible for breakdown of bone. So in nature, you always have checks and balance. So the checkmate for RANK ligand is a protein called OPG, osteopatogerin. So what OPG does is that it binds to RANK ligand. It prevents excessive differentiation of pre-osteoclast, osteoclast precursor into pre-osteoclast, so that the amount of RANK ligand an OPG that is produced under basal condition is just the right amount. Right amount of bone is broken down, right amount of bone is formed. OPG and rank ligand checkmates each other so that the body is just in a state of homeostasis. So if you disrupt this balance, that is where there's trouble. So it turns out that a number of other cytokines in the body that also kind of influences the activities of these two proteins, IL-1, IL-6, IL-7, MSCSF, IL-17A, and TNF-alpha. They promote the production of rank ligand, they amplify its activity, they suppress OPG activity, they also stimulate the expression of the receptor. So where do these two proteins come from? Let's start with OPG. It turns out that the bulk of OPG in the body is produced by B cells. So B cell is the primary source. 2/3 of OPG that is produced in the body is produced by B cell. And so B cell activity, as you know, is regulated by T cells through this co-stimulation between the two cells through the CD40 ligand co-stimulation. So if you have any disease that affects B cells or disease that affect T cells or the crosstalk between B cell and T cells, it will definitely have impact on OPG production. So the second half of the equation is rank ligand. So rank ligand is normally produced by osteoblasts and the synovial fibroblasts. But if there is disease condition, inflammation, any form of inflammation, whether it's due to infection, autoimmune disease, many cells in the body, a lot of cells, almost every cells in the body are activated and they begin to produce rank ligand. So when you take all of this information together, you have the skeletal system, you have the immune system, and you have this convergence of cells and cytokines that plays different roles in these two systems. So this convergence, so we use the word, the immunoskeletal interface to describe this convergence. So got back to HIV. We know the father or the mother of immunodeficiency is HIV. So we talk about CD4 cell. So HIV, you know, the central piece of the adaptive immune system is virtually depleted by HIV. But TCD8 T cells are also affected as well as B cells. We never talk much about the impact of HIV on B cells. You have upregulation of the immature transitional B cells. The activated mature B cells is also upregulated. The memory B cells are decreased. So the question in our mind then is that if cells that produces these proteins that regulate bone metabolism is produced, are the immune cells, and the immune cell is affected by HIV, it's just simple that HIV will have impact on the skeletal system. So the question is that how does that happen? So we decided to explore this a little further. We knew we couldn't start away, if we're going to prove this as a concept, we couldn't start with human studies because human, we have a lot of co-founding factors, things that will affect the bone. CKD, hypogonadism, vitamin D deficiency, alcohol use, drug use will just confound the result. So we decided to use the animal model, the HIV transgenic rat model. So this model has what we call the GAG pol-deleted HIV provirus. They do not have actively producing virus, but they express most of the protein that you will see for the HIV infection. And so they have the same phenotype. Many of the manifestation of AIDS is seen in this mice, like cataract, neurologic symptom, respiratory distress, wasting syndrome. So the question we had was that, do we see the same bone phenotype with these mice, this rat model that we see in human? And so to answer that question, we did something that appeared to be very simple. We did DEXA scan. We took these rats, we did DEXA scan, we did micro CT, and we did plain histologic H&E staining. And what you can see here very clearly is that in the HIV transgenic rat model, you see significant reduction in bone mineral density. you see loss of both trabecular and cortical bone in the HIV model. And then you also see the histologic staining where you see loss of mineralization and the trabeculation. So the next question, therefore, is that what is driving this? Is it because bones are not being formed, or is it because too much bone is being broken down? So when bones are broken down, the body usually secretes this very sensitive marker of bone breakdown called C-terminal telepeptide of collagen, CTX. When bones are formed, they also produce another protein called osteocalcin. So you can measure this protein in the serum to really look at the level of bone formation and bone resorption. And so that is what we did. And so what we found was that, look at C-terminal telepeptide of collagen is markedly upregulated in the HIV transgenic rat, which means that resorption is markedly upregulated. The other thing that was surprising was that formation was not changed, which is unusual because usually when you increase resorption, there's compensatory increase in formation. So you see uncoupling of the process of resorption and formation. So we asked the question, what is driving um, this? Was it rank ligand or OPG? Remember, rank ligand OPG plays a role in this. And we find out that rank ligand is markedly overexpressed. OPG is significantly underexpressed. The ratio of rank ligand to OPG is tilted in favor of excessive osteoclastogenesis. That is, the bone was just, environment is created in such a way that it favors you breaking down more bone, and then you are not able to form new ones. And so it turns out that we then asked the question, which cells in the immune system is doing it? We did immunomagnetic separation, and we found that this was being driven by B cells. A lot of these activities were happening in B cells. So the way we then went back to human, now that we know what we're looking for, we've been able to map the pathway in animal model. We went back to human experiment, did exactly almost the same thing. We controlled for all of the confounding factors like the age, sex, comorbidity, and we came to the same conclusion. There was decreased production of OPG, increased overexpression of rank ligand. All of that leads to rank ligand, excessive bone breakdown. So then kind of the way we summarize this was that in the presence of HIV infection, B cell function is altered, T cell function is also altered, the crosstalk between B cell and T cell is also altered. So all of this leads to abnormality in the function of B cells. B cells is not able to produce OPG and rank ligand the way it's supposed to do. And so you have more rank ligand floating around, not enough OPG. So the ratio of rank ligand and OPG then favors excessive breakdown of the bone. So this is just really the beginning. This was the work we did like 15 years ago. And we've extended that. We've also been able to show that not just the virus, that the drug also do this, but they do it through a different mechanism, through immune reconstitution. We also show that there was a number of nuances. When you age, the impact of HIV on bone loss, the natural process of bone loss with aging is accentuated. And then male and female do not lose bone at the same time. And this process is also altered in men and women. And then lastly, not everybody. So this is kind of, not every HIV infected individual loses bone. So there are some factors that really make some people lose bone than others. And we were able to also show that this is not a phenomenon that is unique to the bone only, that the rank ligand, rank OPG axis affect a number of other organs in the system, not just the bone, immune system, the skeletal system, cardiac system, smooth muscle system, which is a different talk altogether. And we were also able to show that we can block this pathway. So using a repurposed drug, this is a bisphosphonate, zoledronic acid. Given a single dose of zoledronic acid, we were able to completely, so the blue is intervention, the red is control. We were able to completely block bone loss in the setting. And the point I wanted to make with all of this is that during the course of this, we were able to put together a large domestic biomedical research infrastructure. No, by the time I was leaving Emory last year, we had a team of basic scientists, clinical scientists, data scientists, community social workers, and uh, the team was over 150 people. Our research budget was between 15 and $20 million a year to really study some of this phenomenon. And so it was at this time that we started receiving invitation to really do some global work. And so we thought that with the amount of infrastructure that we have built domestically, we can share some of this experience with our colleague in resource limited setting. So opportunity came up in West Africa, in East Africa, Addis Ababa, in Mexico and the Philippines, and then collaboration with the University of Bordeaux in France. And in West Africa, we had about 8 different countries. So today I'm just going to focus on our work in Lagos, just for the interest of this discussion. And the principle of engagement for us was really simple. Partnership based on mutual respect and benefit, leverage the collective strength of both sides of the divide, and ultimately transfer ownership to the appropriate group within the partnership. And so in Nigeria, the first thing, when our collaboration started, I was invited in 2013 to serve on a national committee in Nigeria to revise the medical school curriculum. So they wanted to revise their medical school curriculum. So I was invited to be part of that. And the director of the Nigerian Institute of Medicine, of Medical Research, was part of that committee in Nigeria. So when we met, he wanted to develop some collaboration. Essentially, what he was actually looking for is opportunity for us to provide support. You know, what are the things, can you support some of the things that we're doing? What are the microscope or things in your lab that you're no longer using that you can send to us? Kind of more like a hand-me-down situation. So I told him, what exactly are you trying to get to? It's like, yeah, we're trying to build our research infrastructure. And I said, well, what you need to do is not really look for handout. Let's look at where the needs are. What exactly do you want to build? What do you want your research infrastructure to do? And so we started looking at some of the needs in the country. And they said, it's tremendous. But one area that they identified was just issue of maternal infant mortality, that healthcare for women in the country is just dismal, and we don't know what to do. And so we started this partnership with the Institute. Then we added University of Lagos, which is one of the major academic centers in Africa, and then APIN Public Health Initiative, which is one of the largest CBO, public health, non-governmental organization, NGO in the country. And so after the discussion, I kind of came to the conclusion that what will be helpful is not for us to send handouts to them, but to actually help them build their capacity, to train their people to be able to do the things that they are trying to accomplish. And so to do that, we knew we needed resources. It took us about 5 years of back and forth, and we decided to apply for a trading grant, because what they need was that they know they were deficient. They know they had problems, and they wanted to solve that problem. And we felt like it would be good to empower you to solve your problem than for me to come and help you solve your problem, and then when I leave, you're not able to do anything about it. So we started the training program. So this is our grant number. You will see here, I kind of deliberately left this A1. So A1 means that we submitted the grant. It was not successful the first time. And so we were told, oh, well, good try, not good enough. And why was it not good enough? They said, you don't have enough NIH-funded investigators in the country where you're going to work. And then we rebutted and said, but that is the reason we are wanting to do this. They don't have enough, there's no capacity there. We want to go build capacity. If you wait for the capacity to be there, nothing can be happen. Anyway, we made our argument. They allowed us to move ahead. And so we focus on 3 main things: research methodology, analytical skill, grant writing, scientific communication. And so this is our training plan. So Anna Nicole, who actually is one of the few people leaving Emory to join us here. So she's joined my staff. I have a small skeletal, small N of 2 or 3 research team here at CASE. And so we started this training program. And so what we did was that we brought their Nigerian scientists, we entered them into this, what we call the ENRTP training program. We trained them on research methodology, leadership skills, equip them with core translational science skills, and then also everybody had a hands-on mentored research project. And there was a deliverable. You have to, at the end of the training, which is usually about 2 years, you have to deliver 1, a manuscript, 2, you must submit a grant application. It was necessary. You have to actually, we made you sign a contract that this is what you have to do if we're going to invest our time with you. And these were some of the competencies that we wanted to inculcate into this our training. Many of them, they were domain experts. They knew what the problem is. They know they've been trained in some of the science. What was missing was all of these other qualities. Rigorous researcher, team player, boundary crosser, process innovators, skill communication, as well as systems thinker, people who can think from one system to another, not just walk in their small silo. So we came up with this curriculum, which I'm not going to read through, but just to let you know that we were very deliberate and got our people trained through that process. So this is just a snapshot of the outcome after 5 years. So by the end of 5 years of this effort, we had trained about 28 people. This slide was put together towards in 2023 when we were doing our recompetitive renewal. 23 men, 64% women, 36% men, and look at the number of publications, 53, 150 publications, 3 grants, 30, all of them, 30 of them have, more than 30 of them have obtained grant, 3 NIH grant, 3 of them in the NIH supported mentorship program. And more importantly, 90% of them stayed in the country and they were doing work. So this is a profile of one of our trainees. So Ifeoma Idigbe was a research scientist in the Institute, got into the program in 2020, and look at her publication records. These are PubMed reference publication within the period that she was in the program. And so this is a picture of all of us together, just telling the story of all of us. So the next part, which I'm going to go through very quickly, is that what are these people doing now in the country? What did we train them to do? What are they doing now? The impact of this training. So the thing we wanted to avoid was the white elephant projects. Things like to train them in all of this fancy single cell technology that they would never be able to use, they won't have the resources to use in their country. So the example we gave to them was this. You all know this, if you do medical history, you know Ignaz Semmelweis. Ignaz Semmelweis saved a lot of life by just simple observation. So this was in the 1800s. He was a Hungarian in practice in Vienna. Before he came to Vienna, the delivery of women were done either by the midwives or by medical students. So the midwife would go in and deliver on Monday, Wednesdays, and Fridays. Medical students would do Tuesdays, Thursdays, and Saturday. So medical students and faculty, physicians in general, they'll go to the cadaver room. This is 1800, this 1840, 1850, this idea of microbes, germ, was not part of medicine at that time. So they will leave the cadaver room, they'll go into the delivery room. People didn't wear gloves, deliver babies. Many of the women had puerperal sepsis and they died. This is mortality rate. And then on the days the midwives who are more careful, wash their hands, they deliver baby, no puerperal sepsis, no fever. And so the women became smart. This went on for many years, so that to the extent that when a woman is pregnant and they go into labor, for say on Thursday, they will not go to see the doctor. They will wait until the following day, until it's the midwife's day before they go to see the doctor. So Semmelweis came to Vietnam in 1846. And what did he observe? This high rate of puerperal sepsis. He didn't know what to do. It's like, well, maybe there's something about the cadaver, the smell, and everything that happens when when the trainees go from the cadaver room to their delivery room. And so he put a chlorine bucket, the chlorinated water bucket by the delivery room, and asked all medical students to wash their hands. And that was how reduced puerperal sepsis and maternal mortality by almost 80%. So we told them, this is the kind of thing. You don't have to have this big MRI or this heavy equipment to make a difference. What are the issues? What are the health issues in your community? Can you come up with a practical way to solve them? So that was kind of really the orientation that we gave to them. So we're going to look at maybe 2 or 3 of those projects that these people have taken on. So the first one I wanted to highlight is this CHESS project. I'm sorry that my slides are messed up, but it's really looking at HPV-related cancers. So cervical cancer was a big killer. People die, women, young women especially, died of such. So the incidence is high, so is the mortality. 100% of, almost the death rate is 100%. You get it, you die. And so what we did was, Lisa Flower was my colleague at Emory, a well-renowned OBGYN surgeon, mentored Dr. Ogunshola, who is back in Nigeria. And so what they wanted to do was to figure out a practical, implementable, cheap way to really reduce the rate incidence of cervical cancer. So they took this simple approach that is known to work. So you get swab and you get women to take their swab. And the swab is then sent to the clinic where they do point of test HPV testing. And then everybody in Africa have a smartphone. And so they can actually hook a small device into the smartphone and actually do a speculum exam. The nurses can do that and take good pictures of the cervix. And then they can send this picture to an OBGYN surgeon who sits in a place. You can just download them in the computer. OBGYN surgeon can look at it in real time and tell you there's a lesion in position 8 or 12. And then the women also have this device to do thermal ablation. So very simple. So there's not enough OBGYN surgeons to examine every woman. Women don't have money to go through all of the fancy things that we do here, but here is a practical approach where they can self-collect their samples, where we can do HPV testing in real time. Nurses can use their cell phone to take pictures, and they can actually do therapy. So when we last met with the team, they had HPV positive lesions. 35% of the women that they have screened has lesions, HPV-related lesions that were treatable. They were able to treat about half of them, another half that is awaiting treatment, people that are still awaiting diagnosis. But what was impressive was that the vast majority of this HPV were not the typical HPV that we see here in the US. So serotype 16 was only just about 30, serotype 18 and 45, 53. 80% of all of these lesions were those others. And so that raised the next question. So at this time, the country, Nigeria, was beginning to introduce HPV vaccine. So the question is that HPV vaccine, so we have, as you know, we have about 3 of them. You have the 9-valent, the quadrivalent, and the bivalent. So Nigeria is a resource-limited country, so they were only able to roll out the bivalent, which is cheap, and then they are now introducing the quadrivalent. But if 80% of all the HPV serotype are not in the vaccine, is the vaccine going to be effective? So Chika, who is one of our training, came to Emory, spent some time she's in molecular virology, and is gone back home. And so what he's now doing is collecting all these lesions and trying to look at what type of HPV serotype is driving cervical cancer in the country. Is there cross protection from the strain that are present in the vaccine that are being given in the country? So the next big project was the Salman project. So remember, we talked about maternal and infant mortality. So the rate of infant death in Nigeria is 67 per 1,000 deaths. So to put that in context, in many of the advanced Western countries like Norway, Sweden, Japan, it's about 2 deaths per 1,000. In the US, it's about 5 to 6. Here, in a country where you have close to about 80 million deaths a year, you can do the math, how many children are dying every year. And so 2 of our trainees now took this on as a challenge. And so there has been data that shows that if you give a single dose of azithromycin once or twice a year, you can actually reduce infant mortality by about 25%. So they took on this project, I think, sometime in 2023 in 3 states as a pilot project where they administer azithromycin to children ages 1 month to 11 months. They did about close to 30,000 infants, and they saw signal: 1, it is safe; 2, it did not drive antimicrobial resistance; and 3, it actually is signal towards reduction of mortality. So towards the end of 2024, they got additional money up to $16.5 million grant from the Gates Foundation to roll this program to 11 other states within the country. So this project is ongoing to see if we can reduce infant mortality in the country. This is another project about oral health. So Kehinde Umiezedike is a professor in the Department of Dentistry at the University of Lagos, where in Africa, oral health is completely neglected. So the question is that in people living with HIV, does periodontal disease actually drive the occurrence of non-communicable diseases? And so that is the question that this study is designed to have, because we see hypertension, cardiovascular disease, bone disease in this population. So the question is, do we, through inflammation, because periodontal disease leads to persistent inflammation. In the presence of HIV, you heighten the inflammation that is already due to HIV. Can you drive this process of inflammation and therefore increase incidence of non-communicable disease? So this study was funded through an R01 mechanism, the first R01, I think, for the University of Lagos, and it's currently ongoing. So there are 3 of our trainings that have a Career Development Award, what they call K43. Kehinde Okunade is an OBGYN surgeon who is trying to develop, improve a technique for diagnosing cervical cancer in Africa, cost-effective technique. Dr. Adisa, who is directly mentored by me, she's actually going to be coming here next week as part of our mentorship. She's currently at Duke. She's a biochemist, is looking at ways to diagnose HPV, Hep C, Hep B related hepatocellular carcinoma. And then Hebasin, who is an MD, PharmD. PharmD is looking at the pharmacoepidemiology to understand predator and impact of low-level viremia in people living with HIV. And there are 6 of the other scholars that have received NIH-funded international mentorship program. We've also been able to link some of their leaders to also have a voice in the global scientific discourse. This was the Vice Chancellor of University of Lagos, Professor Ogunshola, who was invited to be the keynote speaker for the 2023 ASM conference. You know, ASM is the largest biomedical conference in the world. About 30 to 40,000 people attend this conference, and there she was trying to share her voice, lend her voice to the scientific discussion. Here is the cumulative PubMed reference. Adaya, who is also with me here now, came from Emory with me, helped put this together recently, and altogether, almost 713 PubMed reference publication, an average of 20 publications per trainee. But what is more important is the impact of what these trainees are doing. It's not just publication for the sake of publication, but what are they doing? Are they changing the course of disease in their country? So we think we're driving innovation. And some of those experiences is what I hope that will bring to our global health effort here at UH and Case Western. A lot already has been done, and you can recognize these faces: Dan Simon, Henry Boone, our own Keith Armitage. They've been to Uganda a number of times, and there's a lot of investment effort to really grow global health initiative here. And our goal is really for me to join my effort with their effort to grow the Roe Green Center to become a world-renowned global health center where a lot of impactful work that changes the curve of disease is happening. We also hope that there will be a multiplier effect where the investment that Roe Green puts into this effort is being multiplied by external grants. And we want robust scholarship with visiting professors as well as endowed professorship. So we do have a lot to do. We're hoping that the Roe Green Center will be something that will really help us coalesce our global health initiative so that trainees, faculty, there could be exchange of ideas on both sides of the globe. So just to conclude, innovation at home has the potential to transform biomedical research effort abroad. But to achieve this goal, I think global health, especially in resource-limited setting, needs to be re-envisioned with a dedicated focus on building local capacity. We didn't go there to extend my research project. I could have gone there for them to collect data from me to come and enhance my work here. But the idea is really their capacity and their capacity to address their local health needs, the priorities of the country. What is it that is bothering the country and how are you solving those processes? And then, of course, leveraging local resources. What we see is that we bring all this sophisticated equipment, they all break down, and as soon as we leave, that work perishes. So with or without us, I think the group that we left behind in Nigeria, the work continues. And so with that, I just wanted to say thank you to everybody that have supported this effort. It's a lot too many to mention, but funding sources, and I'm glad to be part of this amazing community here at UH and Case Western. Thanks again for your attention.
A couple of comments, we'll take questions.
Yeah.
One is, I promise that Henry and I will try to find pictures that are less than 15 years old. But no, I want to just thank you so much for acknowledging Roe Green and Bob Saw at the beginning. And Bob meant so much to us, and I think you're perfectly poised, the synergy of your background and your leadership of our department to extend his work, and we're super grateful for that. And it was a fantastic presentation. Seeing a patient in a clinic, taking it into investigation, and then projects internationally. As you know, we're involved in interviewing lots of intern applicants, and many intern applicants have interest in global health. A lot of residents have interest in global health. And I think sometimes it's hard to conceptualize as a young trainee or young student. How do you go from an interest in global health to sort of what you've done? I don't know if you have 2 minutes of advice or reflection. So yeah. So,
yeah.
And for the Zoom, yeah.
Yes. So the way, again, why are we interested in medicine? Question number one. And then why are we interested in academic medicine? And so that is, medicine is one thing. Academic medicine, you take medicine to that level of curiosity. The reason I'm not out there across the street seeing patients and making as much money as I can make is because academic medicine allows me to exercise my curiosity. I tell you, that patient, I still think about that patient till today. I met that patient in 2005, the same patient, thinking about one patient. How can we make life different from anyone that looks like her, that present with that same problem? That is what distinguishes academic medicine from medicine in general. We approach the care of patients with the heart of innovation. And if you have that, there is no bound to where it will take you. It will take you to the Philippines. It will take you to Bordeaux. It will take you to Lagos. It will take you to Vietnam. If you have curiosity, that curiosity will lead you into different many ways. And I tell people that is really what separates academic medicine from medicine in general. This drive for innovation, to do things different, to do things better.
I'm super excited about this vision. I don't know if there's time for questions or comments from the audience, or I don't know if our colleagues are monitoring the chat. I could grab my laptop and see if there's. I guess I could look up. Uh, okay, Doctor, and we have a microphone. Maybe Dr. Adams, because I'll spaz out. Someone will go, okay, boomer. It turns on, yeah.
Wonderful lecture and a wonderful preview of what can be done worldwide. I guess the one question that I have is, can any of these, does this provide any hints for what can be done locally in Cleveland, Atlanta, for example, or have you been able to expand in any of those strategies for improvements locally? And do you perceive the ability the potential to do that here in Cleveland?
Yeah. Thank you, Dr. Jackson, for that question. So there's this saying, I don't know if it's common here, that charity begins at home. A lot of what we did overseas was because we had done them in Atlanta. So we had a robust team in Atlanta, not just a team of researchers, but we also have connection. We were in the hood in Atlanta driving our research, trying to find solution. I can spend a whole day. We can do a workshop on, neurocognitive disease, cardiovascular disease. Some of the things we find out that not even in HIV patient, that even in our inner city patient without HIV, as control, they came in for a study for control. And we find out that within a period of 2 years, there was accelerated plaque buildup. When we did sensitive MRI of their coronary vessels. And so a lot of this work that we did overseas were informed by what we were already doing at home. So I can give another talk on our domestic effort. Yeah, absolutely. And I'm hoping that here in Cleveland, essentially, as a chair, my responsibility is to work with the leadership of UH to provide healthcare for this community. This community is now my primary responsibility. And I'm hoping that all of us will do our best. We're already doing it, but we will continue to improve, innovate, to make healthcare more accessible, to make healthcare more efficient for our community.
And are any of the residents from the right team here? Because that's Dr. Jackson, right? That's the namesake. So had to point that out, Jackson. Yeah. I know that I think chat, we don't, the chat was disabled, or I got a text from Dr. Robert Bonomo, of course. I don't know if there's anything else in the chat or any of it is after 1 o'clock. And any other comments and questions from Kulos or anything urgent from the chat? No. If not, it's 1 o'clock. I'm going to thank you. Fantastic presentation. We are so glad you're here to lead us. So thank you.